T-shirt say teacher with two E's?
No, it does not. Yeah.
Okay, good morning, everyone. Do you want
I just wanted to thank one person.
Okay, Mr. D's going to thank someone.
I just wanted to thank Ms. Korn for her help at the exhibit. She really saved our butts at the front of the school, and it also gives me an opportunity to embarrass this one right before her presentation. So, thank you.
Okay, so today we have Jen presenting on the, a possible link between stress and schizophrenia. Jen is a first-time presenter, so that means you can ask clarification questions, but no, like, super hard questions, no Andrew Rodesca questions. So here's Jen.
Hi. Okay. My paper is titled Phenotypic Effects of Repeated Psychosocial Stress During Adolescence for the Schizophrenia Risk Gene Neuroregulin 1, a putative model of gene times environment interaction. Or in simpler terms, how stressful social environments interact with existing gene mutations to bring about schizophrenic tendencies. So what is stress? It's a response to a perceived threat. Your body goes into survival mode, and it stops kind of thinking about anything else. It stops being hungry or sad or hurt, and it focuses on surviving the stressful situation. Stress is caused by work, relationships, money, life changes, anything big that requires your full attention that is threatening to your well-being. So when you're stressed, the hormone cortisol is produced because adrenaline's rushing your body, and with that becomes a hormone. But prolonged production of that hormone can be really detrimental to your immune system and your memory and a bunch of other things. If you look in this chart, that's listing a bunch of negative side effects of too much stress. We know that short-term stress is good, it motivates you, it gets you going, but long-term stress affects your physical well-being. It causes pounding heart, high blood pressure, inability to concentrate, irritability, dizziness, things like that. So separate from stress, what is schizophrenia? It is a psychological disorder affecting about 1% of Americans. So there's 320 million people in this country. That means 3.2 million people are living with schizophrenia. It's a pretty equal opportunity disease. It affects equal numbers of male and female and people of every race. So it affects everyone. And its cause is unknown, and because of that, treatments of schizophrenia really focus on suppressing its symptoms instead of kind of like curing a person with schizophrenia. In addition, it's really hard to diagnose because it is a psychological disorder, which means that the only way to diagnose it is by listening to someone tell you their symptoms and using that to diagnose schizophrenia. And the study I looked at in my paper was studying mice with schizophrenic tendencies, and that's even harder to diagnose because a mouse can't tell you, like, hi, I hear voices in my head. So symptoms of schizophrenia include hearing or seeing things that aren't there, lack of pleasure with life, depression, lack of desire to try new things, kind of a decrease in curiosity, trouble focusing, short-term memory issues, aggression, hallucinations, thought disorders, behavioral problems, The study that my paper went into was looking at a link between neuroregulin 1 and an adolescent stressful social environment. So neuroregulin 1 is a regulator of normal neurodevelopment processes, including ones involved in regulating your anxiety-like behaviors. And it's been shown in past studies that there's a certain mutation of NRG1 that is linked with schizophrenia. So schizophrenics tend to have this mutation. They wanted to look at why. And they were going to expose mice with and without this gene mutation to adolescent stress. And they wanted to test the effects of that. They had 4 different groups of mice that they were using to kind of test against each other in the study. There were 2 groups of mice that had this mutation and 2 groups that did not. Out of the groups that had the mutation, 1 group was exposed to stress, 1 was not. And then 1 group of the mice without the mutation was exposed to stress, 1 was not. So 4 groups total. And the way they exposed these mice to social stress was using a known paradigm of like chronic social defeat. So it's a form of social stress. they would put aggressor mice alone in a box for 3 days to kind of allow them to establish their territory. And then after 3 days, they would place the mice that were going to be tested in the box alone with these aggressor mice. And the mice being put in were adolescent mice. They were about 35 days old. They were put in these boxes with the mice, and it was allowed for social defeat to occur. So that's when the aggressor mouse kind of bullies the test mouse until it stands on its hind legs with its forepaws down and its ears retracted. And that's what's defined as social defeat in mice. So they waited for the aggressor mouse to defeat the mouse, and then they would put up this kind of partition between the two mice so that they could see each other and smell each other but not interact. And that partition, and their mice were both present on either side of the partition for 24 hours. And then it was repeated, the 10-minute social stress. And this social stress defeat was repeated for 10 days straight. So it's chronic. It's a repeated situation. And at that point, it becomes that kind of level of negative stress that I talked about before. Too much stress. Yes?
Why the partition? Why not just keep them?
They wanted the stress to stop. They wanted the social stress to stop, for it to not be a constant situation. So they exposed stress, exposed social defeat, and then were kind of alone for a while. And they would switch up the aggressor mice so that there weren't habits forming. So 2 days in a row, it would never be the same aggressor mouse used.
I'm guessing, and I'm not sure you can answer this, but I was just, why wouldn't they put them in another cage? Why wouldn't they separate them?
I'm not sure. So to go about testing how stress would affect this mutation to or to not bring about schizophrenic phenotypes, the scientists put the mice through a lot of behavioral testing to test for certain known phenotypes of the disorder. So this one is the novel open field test, and that is testing for the known phenotype of a lack of exploratory spirit in middle schizophrenia, kind of a lack of curiosity. And they're testing for that using this one. So you put a mouse in a big white box or a big open area, and you test how much they move around, where they move to, and the speed at which they move. So the data I'm looking at right now is the distance they covered while in that big white box. And that shows how much they're exploring and their exploratory spirit. And it's been shown in past studies that for some reason NRG1 mutants do have a higher level of activity in novel open field than if you don't have the mutation. So the white groups are the control groups who were not stressed. And the black groups are the groups that did experience stress. And these are the wild type. These two show the wild type, meaning the mice who were not, who did not have the mutation. And HET stands for heterozygous, or mice with the mutation. So see here, this white bar is higher than this white bar, just because that's something that is seen in NRG1 mutants. And when you're exposed to stress, there wasn't much change in the wild type. Those without the mutation didn't really experience a difference in the way they were exploring their surroundings at all, stressed or unstressed. But the mutants saw a significant decrease. Beforehand, they were covering over 5,000 centimeters in that box. And then afterwards, it was less than that. It's a significant change, as you can see. So that shows that the NRG1 mutation plus a stressful environment caused to bring about that schizophrenic phenotype of a lack of curiosity. And then if you look over here, this measures their fecal excretion, or how much the mice were pooping while they were in that box. So there's not much of a change in wild type, especially with the error bars. It doesn't really change much how much they poop while in the box, stressed or unstressed. Doesn't change much. But if you look at the mutants, there's a significant change in their levels of fecal excretion, stressed versus unstressed. When you're a stressed mouse with this mutation, poops a lot more while traveling around in that box than a mouse with that mutation that is unstressed. And that hints at the fact that the NRG1 mutation makes their autonomic responsivity more susceptible to the effects of chronic social defeat. And autonomic response is the section of your peripheral nervous system responsible for influencing your internal organs. So that's saying basically that the mutation makes your internal organs more susceptible to that stress, making the mice poop more. So they're stress pooping, and that's not good. behavioral testings that they ran were spontaneous alternation and agnostic behaviors. Spontaneous alternation is a way of testing a mouse's spatial working memory. So that is right here. It's the Y-shaped chart. So you put a mouse in a Y-shaped field and you test how often it alters between the legs. So if it goes from leg B to leg C to leg B to leg A and alternating. And that's testing your spatial working memory in that if a mouse goes to leg B, comes out, and then forgets it was in leg B and goes back in, it shows that they forget they were just there. So if you look at the white control groups of the wild type and the mutants, they had pretty high levels of alternation. They were about 45 to 50%, and they were equal, which shows the mutation itself does not affect your spatial working memory. However, once you expose them to stress, there is a decrease in wild type, but not as significant as the one with the mutation. There's a large decrease in your spontaneous alternation when exposed to stress combined with that NRG1 mutation. And that's a revealing of the schizophrenic phenotype of short-term memory loss. Spatial working memory is declining. And then below that, if you look at the agnostic behaviors, which is testing the aggressive behaviors of a mouse towards other mice. So rearing at them, biting them, things like that. There's a dramatic increase in both wild type and heterozygous mutants. And that just shows what we already know, that stressful situations make you grumpy, they make you less sociable, and they affect how you interact with others. Any questions? Aside from the behavioral testing I talked about, they also looked at blood samples of the mice to test for different proteins or hormones and things like that. And in particular, I'm talking about they looked at the corticosterone, because corticosterone is a steroid hormone involved in regulating stress, energy, as well as immune reactions. So regulating energy, that hyperactivity that we saw in the box, the immune reactions, the fecal excretion charts, and the stress regulation. So corticosterone levels are heightened in both wild type and mutants when exposed to stress. And I think that's important just because it shows that stress has a greater impact than just how you're behaving. It impacts your blood. It has a deeper biological change than just how you're temporarily feeling. So that's what people should keep in mind when they realize that they've been stressed for prolonged periods of time. So what did the scientists kind of take away from this study? They took away, they kind of established for the first time a role of adolescent stress interacting with NRG1 mutations to bring about schizophrenic phenotypes. So it's been known that NRG1 mutation is linked to schizophrenia, but it really was unclear why. And now it's kind of hinting at the fact that maybe a prolonged stressful environment in your youth combined with a pre-existing mutation in that gene could bring about the tendencies. Because if you look at like this graph right here, spontaneous alternation, there's no difference between the mutants and the wild type to begin with. It's not the mutation itself that is bringing out the schizophrenic tendency of spatial working memory. It's schizophrenia combined with stress. So that's a new place they're taking schizophrenia because it's so hard to diagnose. They've been looking at why the mutation is involved, and like this is a new direction to take the study. And then the bigger picture, aside from this study in particular, is epigenetics. So epigenetics is when forces outside of your DNA change your gene expression. And it has to do with maybe your environment, your age, your lifestyle choices, etc. So it's no change in your DNA sequence. Maybe your gene expression is changing because you're 70 years old, or because you're a chain smoker, or because you're exposed to a stressful environment for a really long time when you're young. So specifically in this case, the environment we're talking about is the prolonged stress. And that's going to combine with latent gene mutations of NRG1 and cause gene expressions to change, bringing about schizophrenic tendencies. So as a whole, it's kind of targeting NRG1 mutation as a major part of schizophrenia. And while schizophrenia is so hard to diagnose because it's a bunch of different things rolled into one, and it is a psychological disorder, so it's just difficult to diagnose as a whole. It's just kind of hinting at an indicator of schizophrenia. So if you have this NRG1 mutation, they're going to say, well, like, that's an indication that it's possible that you're going to have schizophrenic tendencies when you're older, should you be prolonged to a really stressful environment as a youth. Or they could be looking at someone who's already schizophrenic and look for that NRG1 mutation and maybe look at it as a cause. So it's just kind of an exciting new direction to take the study of schizophrenia. So I just want to say a quick thank you for my presentation to Cindy and Mark and Amanda, Catherine, Jake, Wesley, Julia, Adam, Jack, Mr. D, Mr. Sullivan, the whole Journal Club team, and for letting me be on the team because I've had a really great year. And yeah, thanks.
So where did they, I don't know if you noticed, but where did they look, or where did they find the pre-existing
Mutation.
Yeah, and where did they?
So I think I kind of am shaky on it, but I think they took them, since they're heterozygous, they had like two parents, and one with a mutation, one without.
Sorry, I meant like, that you said that they had studied it earlier and they knew that there was some
Yeah,
mutation.
in past studies that the scientists were like familiar with.
In mice?
Yes, in mice. Yeah.
Can you like tell me like what kind of period of time they measure like the stress levels of like, the mice?
Yeah, so when they started, they were 35 days old, and then they went through that 10-day stress cycle, and then kind of immediately after that 10-day chronic stress, they tested it. So it was testing the kind of like immediate emergence of these phenotypes, but they were definitely stressing because they wanted to get to the mice while they were still young, because the point was like adolescence.
Next is Wesley, and Wesley will tell you what she's presenting on.
I am presenting on how I'm presenting about how a brain becomes like female or male through DNA methylation.